Hello students, welcome to UGC NET 24/7 and this is your Faculty of Life Sciences. That is for your CSIR NET preparation. I'm sitting in front of you and today the topic of discussion is production of male gamete. यानी पुरुषों में युग्मकों का निर्माण किस तरीके से होता है? What is the procedure involved behind it? Let us discuss. So I think everybody is ready. Yes? Hi Anna. ठीक है जी. Good evening everyone. So first of all, when we are discussing about a male gamete, that means that will be produced by a primary sex organ of male. So primary sex organ of male is a pair of testes, right? And secondary sex organ also known as accessory reproductive organs are vas deferens, then we have epididymis, we have seminal vesicle, prostate gland, and bulbourethral glands. Bulbourethral glands are also known as Cowper glands. Right? So these are the accessory reproductive organs of male. Now, where does this gamete gets formed? So if I take the structure of a testicular lobule, it looks like this. But before that, I'll try to give you a annotation of a testicle first. So this is a testis, and each testis contains 200 to 250 compartments or chambers, also known as testicular lobules. So these are testicular lobules. And they are 200 to 250. Got it? Now within each testicular lobule lies 1 to 3 highly coiled seminiferous tubules. So these are the seminiferous tubules where the spermatozoa will get formed. So these are 1 to 3 highly coiled seminiferous tubules. 1 to 3 highly coiled seminiferous tubules, where formation of spermatozoa will take place. Where process of formation of spermatozoa takes place. Getting my point? And these compartments are called testicular lobules. And this red 1 to 3 highly coiled are seminiferous tubules. These are seminiferous tubules, which are 1 to 3 and highly coiled. बहुत ज़्यादा कुंडलीकृत होते हैं, and they are highly coiled structures. Okay. On the other hand, if you see the anatomy of a testis, it is like this. this is the vas deferens, which loops over the urinary bladder and then enters into the seminal vesicle and forms the ejaculatory duct. Vas deferens ही आगे जाके ejaculatory duct को बनाता है. Now there are minute ductules, पतले पतले ductules होते हैं, and these ductules are called rete testis. These ductules are called rete testis, and then these ductules open into a wider cross section which is called vasa efferentia. That is your vasa efferentia. So this is your vasa efferentia. These bigger columns are your vasa efferentia. Okay, and now they will ultimately reach to a location where these spermatozoa will be temporarily stored. Sir, temporarily stored spermatozoa अचानक से कैसे यहाँ होने लगी? I'll explain it to you. Just give me a second so that I can complete this diagram first. So this is the epididymis where sperms are temporarily stored before ejaculation. So before the ejaculation is happening, sperms are stored temporarily here. ठीक है, this is epididymis. And these temporarily stored sperms at the time of excitement or during copulation, they get ejaculated. After these are transported via epididymis to vas deferens. So these were temporarily stored sperms in epididymis. Got it? Now try to understand. I'll zoom out this testicular lobule, and then you will get to understand what happens within this testicular lobule. So this is a testicular lobule, and within this testicular lobule, we have 1, 2, 3 highly coiled seminiferous tubules. We have 1, 2, 3 highly coiled seminiferous tubules. So it can be 1 also, it can be 2 also, and it can be 3 as well. But the main thing is that they will be coiled. They will be highly coiled. बहुत ज़्यादा कुंडलीकृत होंगे. ठीक है? So this is the seminiferous tubule within a testicular lobule. So this is a testicular lobule. And this is a seminiferous tubule. This is a seminiferous tubule, which are 1, 2, 3 in number. Now within this seminiferous tubules, what happens is sperm get formed. Spermatozoa is formed. These are the spermatozoa जो इस seminiferous tubule में बनती है. I hope it is clear to everybody. हाँ या ना? Okay. Now, we got to understand the location where the spermatozoa is forming. Apparently, this spermatozoa also needs nutrition for its survival. So, where does the nutrition, from where is the nutrition coming? कहाँ से वो पोषण मिल रही है? तो within this seminiferous tubules, we have Sertoli cells which give nutrition to this developing spermatozoons. तो ये जो spermatozoon बन रहा है, इसको पोषण देने वाली कुछ कोशिकाएं मौजूद होती हैं seminiferous tubules के अंतर्गत, which provide nutrition to this developing spermatozoon. I hope it is pretty much clear to every one of you attending the session. So these are Sertoli cells which provide nutrition to the developing spermatozoon. Apart from that, there are some specialized cells called interstitial cells of Leydig. These are interstitial cells of Leydig, which secrete androgens. Androgens को वो secrete करते हैं. And androgens are male sex hormones in which the major or the main male sex hormone is testosterone. The major male sex hormone is testosterone. So you can see the location of Sertoli cells as well as these are interstitial cells of Leydig. Interstitial cells of Leydig. They are interstitial cells of Leydig. And this is a cross-section of your TS of seminiferous tubule. This is a transverse structure of seminiferous tubule in which you can clearly observe the Sertoli cells which are providing nutrition to the developing spermatozoa, and on the other hand, interstitial cells of Leydig are secreting androgens which consists of the male sex hormone or the main sex hormone testosterone. Now this is very much easier to understand that if a particle gets nutrition from Sertoli cells and power from interstitial cells of Leydig, so it won't be stable within these tubules. इन छोटी छोटी कुंडलीकृत ट्यूब्यूल्स के अंदर वो स्टेबल नहीं रहेंगे. किसी को भी अगर खुराक और पावर मिल जाए तो वो एक जगह स्थायी होके नहीं रहेगा. And the same thing happens with this spermatozoon. As soon as it receives the nutrition from these Sertoli cells and power in form of hormone from interstitial cells of Leydig, they rupture this boundary of seminiferous tubules. तो seminiferous tubules की ये जो boundary है उसको वो फाड़ देते हैं. They rupture it. And rupture होते ही ये फिर अंदर के spermatozoa बाहर आ जाते हैं. They move out of this seminiferous tubules. And where do they go? Now after rupturing the seminiferous tubules, they will enter into this small ductules which are called rete testis. So this is what? This is rete testis. And the ductules of wider cross section as compared to rete testis is vasa efferentia. Vasa efferentia. So you can easily now conclude that after it received power and nutrition within the seminiferous tubules from interstitial cells of Leydig and Sertoli cells, from interstitial cells of Leydig and Sertoli cells, what happened is they ruptured this seminiferous tubules, and after rupturing the walls of the seminiferous tubules, they entered into these fine ductules, and these fine ductules are what? They are rete testis. Now this rete testis, if you move across See, I am showing you the TS of testis. ये testis का TS है, ऐसे बीचों बीच काटा है आप, इसलिए आपको दिख रहा है. अदरवाइज यू कैन अंडरस्टैंड कि ये कंपार्टमेंट के अंदर वाले क्रॉस सेक्शंस में हो रहा है, जो फाइनली जाके एक जगह ऐसे मिल रही हैं. Getting my point? So testicular lobules के अंदर seminiferous tubules got ruptured after it received nutrition and power from Sertoli cells and interstitial cells of Leydig, entered into small ductules called rete testis. From rete testis it migrated to vasa efferentia. These are ducts of larger cross section, and finally these spermatozoa will travel down to epididymis. Now what is epididymis? So it is a temporary storage before ejaculation. It is a temporary storage of sperm before ejaculation. And what is this? This is called epididymis. At the moment when copulation is about to happen, after some time the couple gets excited and there is a point of excitement where the male starts ejaculating. So जैसे ही वो ejaculate करने जाएगा, the sperms which are temporarily stored in epididymis, they will be sucked down through this vas deferens and they will reach the ejaculatory duct. I hope you are understanding. So what are these? These are vas deferens. And you might have heard about sterilization technique. So male sterilization, how it is done? We cut this vas deferens, and then we tie it. So पुरुषों में जो नसबंदी की बात करी जाती है, male sterilization, how do we do that? We cut open this vas deferens sideways. This will be this way, this will be this way, and then we tie it. This is called vasectomy. Vasectomy. So male sterilization is called vasectomy. This is called male sterilization technique. In females, it is called tubectomy because we cut the fallopian tubes and then we tie on both the sides. So what will happen? The sperm will not be able to migrate further into the prostate, seminal vesicle and bulbourethral glands where they will receive the alkaline fluid. So I hope you did understood how and where did these spermatozoa got produced. तो ये testicular lobule है. Testicular lobule के अंदर, if this is a highlighter, I guess. Let me shade it in a boundary, then you will be able to understand. Is it visible now? या पहले ही सही था? पहले ही सही था, I guess. It is clear in this way. So this is a transverse section of a seminiferous tubule. Female tubectomy, Diksha, that is female sterilization is called tubectomy, where fallopian tubes are cut and tied. And this is the structure of a testicular lobule, which contained 1 to 3 highly coiled seminiferous tubules. Highly coiled seminiferous tubules ke andar, we could see the Sertoli cells and interstitial cells of Leydig. Sertoli cells was providing nutrition, and interstitial cells of Leydig, they were secreting androgens, or androgens में जो male sex hormone, main major male sex hormone है, that is testosterone. And that testosterone and the nutrition empowered the spermatozoa to rupture this seminiferous tubule. And as soon as the walls were ruptured, it entered further into small ductules called rete testis. From the rete testis, it moved further into greater cross sections which are called vasa efferentia, and from vasa efferentia they migrated into a temporary space where they were stored before ejaculation. I hope it is clear. So this was This was in short, the production of male gamete from testis. And testis is what? It is the primary sex organ present in male. And the sperm, which will further migrate into the ejaculatory duct, it will receive secretions from prostate, bulbourethral glands and seminal vesicle. These organs which support in the transport and forms the passage of this gametes, they are called secondary sex organs, which are vas deferens, epididymis, seminal vesicle, prostate, bulbourethral glands and Cowper glands. I hope it is clear to every one of you. Yes. Chalo. So I'll come up with some more important sessions on this topic. Tomorrow we'll be discussing production of female gamete. But there is one more update from tomorrow onwards, effective tomorrow. Effective tomorrow. आज तो हो गई session. So effective tomorrow, your regular CSIR NET Life Sciences classes on YouTube will happen on a new timing, that is 11 a.m. Monday to Saturday. So Monday to Saturday 11 a.m. होगी CSIR NET Life Sciences की classes कल से. So the timing was 4 p.m. in the evening, but due to some unavoidable circumstances, the timing has been changed to a new timing, and that is 11 a.m. every day on UGC NET at 24/7. So see you again tomorrow, where we'll be discussing production of female gamete. That's it from today's class. Take care of yourself. Do share and watch the session completely. Thank you.